So the next presentation is by Rachel Gibbs, a master's student at the Institute of Medical Science, whose talk is titled Rebuilding the Brain's Lost Cells.
Imagine you're fatigued, you can't focus at work, your legs are weaker than usual. This is the reality for someone diagnosed with multiple sclerosis, or MS, and eventually they may lose the ability to walk or speak because their brain cells are dying. And while treatments can slow disease progression, they can't reverse this damage. That's why I'm studying a new strategy to tell the MS brain how to repair itself. Now, our brains are made of billions of cells. We have neurons. They're the wires which communicate with electrical signals. We have astrocytes. In the healthy brain, they're a jack of all trades which keep everything working. And we have oligodendrocytes. These insulate neurons by surrounding their long bodies. This keeps electrical signals flowing, like the plastic insulation on a wire in your house. But in MS, the immune system is attacking oligodendrocytes, and astrocytes love to respond to these immune signals. They can become protective and clean up debris, but sometimes they can become toxic, killing more oligodendrocytes. And now the electrical signals in the brain's wires can't flow properly. That's why I want to take those toxic astrocytes and convert them into oligodendrocytes through a process called reprogramming. And what if I told you that astrocytes already have the instructions they need to do this? They just need a push. That's because all the cells in our body are in from the same very large set of instructions, our DNA, but they only use specific sections, giving them different structures and functions, like astrocytes and oligodendrocytes. If your DNA was like the manual to every Lego set ever made, bound into one book, to make, say, a house, you would only use a few pages. But what if you wanted to turn your house into a car? You could flip to that page, maybe you first add wheels, and it would look strange. But as you make incremental changes to the bricks, it would look and function more like a car. To reprogram astrocytes, we deliver a piece of DNA instructions that oligodendrocytes would usually use. This unlocks new sections of the DNA, and they use these instructions to shift towards being those insulating cells. In the lab, I'm growing astrocytes in a dish and activating them to that toxic state by mimicking the body's immune signals. Then I deliver that oligodendrocyte DNA, and I found something really exciting. These toxic astrocytes are very good at reprogramming. This gives us a two birds with one stone approach, where we can remove a cell that's contributing to disease by using it to replace the dying cells. One day, we hope to use reprogramming to help people with MS and other neurodegenerative diseases by telling their brains how to repair themselves by rebuilding lost cells.